Thank you very much for the invitation to present in this webinar. So this is the last talk, and I'm gonna explain and describe a bit the COVID-NMA project, which before Clarice mentioned as a source of their data. And I will focus a bit more on the challenges we encountered during this the process of this project, which is a quite different living review in comparison to the standard living systematic reviews that we are used to, where the data are updated every 4 or 6 months. So once the COVID-19 pandemic broke out, clinicians and guideline developers urgently needed up-to-date and high-quality evidence synthesis to inform their decisions. So far, there are more than 4,000 randomized controlled trials registered, so there was an explosion of research outputs, and that's why synthesis of all this evidence was necessary to guide evidence-based and timely decisions. And we realized that existing evidence synthesis approaches were limited to capture all the evidence in the context of a pandemic, because there is usually an important delay between evidence generation, evidence synthesis, and evidence dissemination. So our objective with COVID-NMA was to develop a new evidence synthesis model for bridging the gap between evidence generation, evidence synthesis, and decision making. And we also wanted to make our findings and outputs quickly available to all stakeholders through a dedicated website that we have created. We have a broad scope. We included studies for therapeutic interventions, preventive interventions, and vaccines. And we see this as a proof of concept model, which can be used afterwards for other conditions. So here, this is the COVID-NMA model, and we can see that there are 3 main pillars. The first one is the living mapping of registered trials. The second one is the living systematic review of published trials. And the last one is the living monitoring and feedback in order to make the connection between funders and trialists. So in terms of living mapping, once every week, there is an automatic search in different registers in order to identify new trials. And then these studies are stored in a data warehouse. The data extraction is semi-automated, so some data are automatically extracted. And then some other data need to be extracted manually. And then once a week, all these new studies are included in the mapping of all the trials that we have on our website. In terms of the living systematic review, every week there is a search for new studies, new published studies in the LOVE platform, which has been developed by the Epistemónicos team. We screen and select the studies. We extract the data, we assess the risk of bias, and there is a quality control in collaboration with the Cochrane bias methods group. Then we update the analysis, we grade the evidence, and then all the results are uploaded on our website. And then the last part is to monitor outcomes, the risk of bias, completeness of reporting, data sharing policies, and contact funders and trials to provide feedback. And all these are available on our websites, which is followed by researchers, funders, regulatory authorities, and guideline developers. And in this way, we aim to facilitate evidence-based decision-making. So this is the website, the COVID-NMA website. Here we have the homepage where we can see some information regarding how many trials registered are existing on the platform, how many studies have been used for the synthesis of the studies. And there are also some other information, such as a summary of main results, which is updated regularly. You can find a bi-weekly update, so what kind of updates there are every 2 weeks. You can register to our newsletter, and you can find information about excluded studies and retracted studies. So an important challenge of such a project is to compromise the validity of the whole process in order to obtain as fast as possible results. And this is something, of course, that we don't want. And that's why in our review, we have followed the good practice requirements in every step of the review. So when we set the PICO for every research question in the assessment of the risk of bias, taking of assumptions before analyzing any data, in the definition of the synthesis model, and particularly in the interpretation of results. Another challenge is that sometimes there might be too much emphasis on statistical synthesis, but we need to be careful for that, because in a rapid review, as also Andrea mentioned in the very beginning, statistical synthesis is not always the key aim. For example, there might be very few data which may make statistical synthesis useless. The assumptions might be implausible. There might be a lot of variation in the study credibility, and this should always be taken into account, particularly in the context of a pandemic, where there are many trials published very quickly. We had retracted papers, which ideally they shouldn't be included in the synthesis. A very well-known example is a study for hydroxychloroquine that raised concerns regarding the safety of hydroxychloroquine, which was why WHO stopped the hydroxychloroquine arm in the Solidarity trial, and then this study was retracted. There are interim results published that are not always consistent with final results, and so all these characteristics should prevent us from emphasizing on the summary effects. A key characteristic of our review was that we followed a living process in all aspects of the review. So usually when we refer to living systematic reviews, we mean the incorporation of new studies in the review and in the data synthesis. However, in our review, every time we had new data and new knowledge about the disease, because that was a very new disease that nobody had a lot of knowledge before, we reevaluated all our recommendations. And an example for that, that many people may know, is that we changed our plan for the conduct of network meta-analysis. So the initial plan was to perform a large network meta-analysis with all possible treatments. And then we realized that it's not the best approach. And so we moved to smaller networks with more homogeneous studies and more homogeneous treatments. So we need to have in mind that network meta-analysis, when there are very few data, might be not so useful, in the sense that you might obtain less precise results than individual studies or pairwise meta-analysis. And also, network meta-analysis, most importantly, relies on assumptions. And if the assumptions are not plausible, the results might be invalid. And the key assumption of network meta-analysis is what is called transitivity, that also Archie mentioned at the beginning, which means that we should have an homogeneous set of studies across comparisons in order to be able to obtain valid results. So sometimes people believe that the most difficult part in the network meta-analysis is to conduct the statistical analysis, which is not quite true. So the most challenging part is all the steps before. So it is considered, for example, that a network meta-analysis could have any type of studies, any type of treatments, any type of patients, and then we just draw the network, we put everything in a statistical model, and we obtain the results. In the COVID-NMA project, we moved away from this approach because the risk for obtaining invalid results was very high, because as new data were coming into the database, the results, we realized that the studies are very heterogeneous, the treatments are very heterogeneous. For example, we had populations with different baseline severity. The standard care was defined differently across studies. There were many co-interventions. So we decided that it's better to move to smaller networks and combine all the treatments with similar mechanisms. And apart from that, when you embark on such a living project, we need to be ready to either to broad or to restrict the scope of our living systematic review, depending on the resources that we have and on which are the most relevant questions. For example, at the beginning of COVID-19, the most relevant question was which are the best treatments for COVID-19, and then this changed into which are the most effective vaccines. Now, in terms of data sharing, that was another key part of our approach. We wanted to make data available, and that's why we, from the very beginning, because we had a lot of requests, we developed a concrete data sharing policy. So first of all the data are freely available on our platform. So this means outcome data from the studies and summary effects, risk of bias assessments, and the most important study characteristics and population characteristics. And in terms of the database, we give priority to guideline developers and other related organizations, because as I said from the beginning, that was the main target, to facilitate the work of stakeholders. So we have shared specific data with NICE, with Cochrane Austria. And then for other types of further research, reuse of data, for example, for methodological work, etc. All the data after every publication are becoming available on Zenodo. So for instance, we have published a review for IL-6 and IL-1 drugs, and this data can be found on Zenodo. And the mapping data are also available through our platform after free registration. And we also share the data through a tool that we have developed, which is an ArtSigning application. And we have embedded all the data for COVID-19 treatments and COVID-19 vaccines. And so the user can freely use our database through this tool. So they can use the database to perform their own meta-analysis. They can have different options about the statistical model. There are different subgroup analysis and sensitivity analysis, which are based on our protocol. And then they can obtain the forest plots that we present on our website, where we always present the results along with the risk of bias assessments. And I will close with some notes on automation. So it is clear that such a project is an extremely resource demanding process, and it's really unlikely to move forward without some automation tools. So we have managed to automate or semi-automate several parts of this living review. As I said from the beginning, the mapping of registered trials is semi-automated process. The LOVE platform has helped a lot in order to reduce the time we need for screening and selecting studies. We have also an automated tool for uploading the studies on the platform. So once the data are extracted and verified afterwards, automatically they appear on the website. Of course, all the statistical analyses are automated. And there are still some quite time-consuming parts, which is the data extraction. So far, it is very difficult to create a tool for automated data extraction. And particularly in case of observational studies, the risk of bias assessment also is quite a time-demanding part. So before closing, I would like to thank all the contributors of the COVID-NMA project. This is actually only a very small list from the full list of contributors. And thank you very much for your attention.
Thank you, Anna. That was great, excellent talk. There is a question in the chat from Catherine. So this is for both Clarice and Anna. So do you have a mechanism for withdrawing studies if the studies prove to be problematic? And if so, is there a link where withdrawn studies can be seen?
So we have, can I answer, or maybe Clarice wants to? Okay, so from our side, as I said, we follow all the comments for studies that So once the study is published, there are after several comments from the general community. So we follow all that. We follow, of course, a retraction watch. So once we identify some problems, we immediately take out the study if there are very important concerns about the reliability of the study. We take it out from our review. And as I said, there is a bi-weekly update at the homepage where you can see the studies that have been removed there.
Clarice, do you want to respond?
Yeah, so on our tool, if you do, because we wanted it to be very much the user kind of decides what they do with it, quite different to Anna's. Yeah, you can just, with that sensitivity analysis tool, you can literally just tick a box to say, what will the analysis look like without that study? I don't know if it would give a link. I don't know quite what you mean there. Maybe that was more for Anna's, but for our tool, yeah, you simply just tick the box and then you'll get the results with it gone.
Okay, thank you. And one more question from Kate. So this question, how did you manage the issue of different iterations of the same paper? So for instance, if you've included an early preprint of the paper, did you update the review once the final revised manuscript was published? And how do you match up and reconcile different versions of the same paper when your searches are automated?
Yeah, is it for me?
I guess so, yeah. I mean, anybody else can answer as well, but if you,
okay.
If you want to, Anna.
So yeah, actually that was a quite important challenge from the beginning, because preprints are also updated. So it's not only the update from preprint to publication, but the same preprint might be updated. So we follow all these updates. We have created tools that can monitor all these updates. And so once there is an update, we also update it in our database.